In this lesson, we're going to take a closer look at the digestive enzymes that are involved in breaking down the food that we eat. So, throughout the digestive systems, there are enzymes that are breaking down the food into useful substances. So, recall that enzymes are protein catalysts, they speed up chemical reactions. The substance that an enzyme acts on is called its substrate. So the enzyme acts on the substrate to create a product. Enzymes are contained within the digestive juices that are secreted into the digestive tract by exocrine glands. An exocrine gland is a gland that secretes substances through ducts or tubes onto a body surface or into a cavity. This is in contrast to endocrine glands, which secrete substances directly into the bloodstream. So the exocrine gland creates something and puts it right into the place where it's needed, whereas an endocrine gland creates hormones, is what it makes, and they go into the bloodstream and travel around to where they cause an effect. And we're going to just focus on exocrine glands for digestion. So the exocrine glands that play an important role in the digestive system are the salivary glands, the gastric glands that are in the stomach wall, the pancreas, and the glands in the wall of the small intestine. So first we'll talk about saliva. So we know that saliva is produced by the salivary glands, and it contains mucus, and the enzyme that it contains is salivary amylase. So salivary amylase breaks down starch, which is the main polysaccharide carbohydrate that we eat. And so it breaks down starch into maltose, which is a disaccharide. Okay, so starch is broken down to maltose by the enzyme amylase. So that starts to happen in the mouth when the saliva is added. Next, moving on to the gastric glands. So the gastric glands in the stomach wall produce gastric juice. And gastric juice contains a few things. It contains mucus, it contains hydrochloric acid, and it contains the inactive enzyme precursor pepsinogen. So we're going to have a couple of these inactive enzymes that are produced as precursors, and then they need to be activated. So the reason this happens is because if they were secreted directly in their active state or created in their active state, they could be potentially harmful to the cells that are making them. So basically, you don't want the enzyme to start working until it gets to the place where its job is supposed to be done. So this inactive enzyme precursor, pepsinogen, is activated by hydrochloric acid, and that converts it into pepsin. And pepsin's job is to convert large proteins into peptide chains. Okay, so a protein is a very large molecule, long amino acid chain that is all folded up together. So pepsin's job is to start to break that down into smaller peptide segments. Next, we move on to pancreatic juice. So the exocrine glands in the pancreas produce pancreatic juice. And pancreatic juice contains bicarbonate ions, which provide the alkalinity, which is neutralizing the acid. And it contains a lot of different enzymes. Two of them are pancreatic amylase and pancreatic lipase. And it also contains another one of these inactive enzyme precursors, and this one is called trypsinogen. So you notice the inactive versions, they have this ogen on the end of their names. So pancreatic amylase carries out the same reaction as salivary amylase. So it's just continuing what salivary amylase started, converting starch into maltose. Pancreatic lipase breaks down triglycerides into glycerol and fatty acids. So you have triglycerides, and lipase acts on them to make glycerol and fatty acids. So we've touched on this in previous lessons, the problem with breaking down lipids. So lipids are insoluble in the digestive tract, which is an aqueous environment, lots of water. So as food travels through the digestive tract, the lipids within them melt and coalesce into large droplets of fat. The enzyme lipase is water soluble. It likes the water, so it can't go right into those droplets and break down the lipids. It can only work on the surface of the droplet. So if that was all lipase could do is just work on the outside of the droplet, then a lot of the lipids would just pass right through our bodies because there isn't enough time for lipase to completely digest them. So this is where bile comes in. So bile molecules have a hydrophobic end and a hydrophilic end. So hydrophobic meaning it doesn't like water, doesn't interact with water. Hydrophilic, it does interact with water. So because it's the type of molecule that has an end that likes water and an end that doesn't, it can interact with both lipids and water. So in doing that, it causes the lipids to break into smaller droplets, and this process is called emulsification. And this speeds up the digestion of the lipids in the small intestine. Okay, the next enzyme that was produced by the pancreas was an inactive enzyme precursor, trypsinogen. So trypsinogen is activated by another enzyme called enteropeptidase. And enteropeptidase is secreted by the lining of the small intestine. And so enteropeptidase converts trypsinogen to trypsin. And trypsin continues the breakdown of proteins. Okay, so Pepsin has already broken the proteins into peptide chains. Now trypsin continues to break them down into smaller little peptides, maybe a dipeptide or a tripeptide. So it continues breaking down the proteins. Maltase is another enzyme that's involved, and it's produced by glands in the wall of the small intestine. So maltase is what's known as a disaccharidase because it breaks down disaccharides. So you might have noticed with the enzyme names that a lot of them end in A's. And so that kind of symbolizes that they break something down. And so maltase breaks down maltose, right? Just like lipase breaks down lipids. So maltase breaks down maltose into two glucose molecules. Okay, so through the action of amylase and maltase, we've been able to break down the starch right down to glucose, which is what our body cells need in order to carry out cellular respiration. The enzymes that are produced by the wall of the small intestine are a little bit different. They're not secreted, which means they're not sort of free floating, put out into the digestive tract. Instead, they remain attached to the plasma membrane of the cells lining the intestine. And their active site, that's the spot where they bind to the substrate, is exposed to the food in the intestine. So you've got, like, as the food goes through the intestine, all along the sides, there are these enzymes that are acting on it. And so with this arrangement, the substrates can be digested and then the products can be immediately absorbed into the body because they're right there at the plasma membrane. There are some macromolecules that humans cannot digest, for example, cellulose. And the reason that we can't digest cellulose is because we don't have the enzyme. So we don't have the gene that produces the enzyme cellulase. So we cannot digest cellulose. So we're also going to talk a little bit about how the secretion of digestive juices are controlled. So the secretion is regulated by both nerves and hormones. So nerves carry signals to and from the brain, and they cause specific responses throughout the body. Hormones are chemical regulators that are secreted in one part of the body, and these are those endocrine glands, and they're transported by the bloodstream to another part where they cause a response. What happens is if you see food or you smell food, the brain sends nerve impulses to the stomach to get it to start secreting gastric juices. And then as soon as the food actually enters the stomach, then that sends a signal for even more gastric juice to be secreted. And this happens because receptors in the stomach lining, they respond to the presence of food, send impulses to the brain. Brain sends back impulses to those exocrine glands so that they start producing all the digestive juices that are required to break down the food. It also sends impulses to endocrine glands in the stomach wall, and they release the hormone gastrin into the bloodstream. Gastrin then travels to the upper part of the stomach where it stimulates increased secretion of hydrochloric acid. So those are the two ways that the secretion is controlled. There's a nervous control through your nervous system, and then there's also some hormonal control causing these digestive juices to be secreted when they are needed, when food is actually coming into the digestive system. So that's our overview of digestive enzymes.